Hello, this is Sarfaraz Niazi. I'm totally delighted to be here with you this evening. My topic today is extremely current and extremely important for all of us. It relates to chemistry as much as it does to the biology. My topic is the new mRNA vaccines and how they can be manufactured in Pakistan using very small investments and capital to meet one of the most significant needs of the country. A little bit about myself. I've been around for a long time, as you can see. Currently, I'm involved with University of Illinois and also my own company making these vaccines. I have provided assistance to many companies, including the FDA and others, to write some of the guidelines. And I'm hoping that today I will be able to share with you some of the important things that you need to understand how to make the medicines of the future. So it was back in October 12th, 1768, when we saw the first vaccine administered. This was a smallpox vaccine. Now, if you look at almost more than 200 years later, we have a new vaccine, which is called the mRNA vaccine. It's a nucleic acid vaccine. It was approved for the COVID. I got my shot, so I'm feeling good about it. No issues at all. Now, this vaccine approval is just as important as the first vaccine ever produced. And the reason is that it totally changes the paradigm. So let me give you a little bit about Mr. Kuhn's theory of what is paradigm shift. We live our normal life every day. The difficulties come, we resolve them, and we move on. And then certainly a crisis comes. A crisis comes for which we have no solution. This was one of those crises. Vaccines take years and years to develop. We didn't have time. So we went through crash testing a product that was already in the pipeline, but nowhere close to being tested as well it was. So it is only when you have a crisis and you can find a solution that your way of doing business in life changes. That's what happened last year, is that how we will make vaccines in the future. And more than vaccines, how we will have new treatment modalities has changed forever. So a quick one about types of vaccine. I'm sure I don't have to tell you a lot about these things. These are, you can give attenuated virus or inactivated, or you can give a part of the body of the virus or the bacteria. And then, of course, you can give a toxoid also. And also now we have RNA vaccine. That is the topic that I'm going to talk about today. So let's look at very quickly, what happens in vaccines, how the immune response comes. Let's say you have a virus on the left-hand side. You can take it, inactivate it, and take it straight into the body, and it will produce antibodies, no issue. But then you can also, you can attenuate the virus, make it weaker, and then take it in there, it will still produce antibodies. Or you can break a piece of it and you put it in the body, you still make antibodies. Or you can put this viral genome code inside another virus, which is safe virus. You put that in the body, it will produce antibodies. But now you have another possibility, is that you avoid all those things. You create the RNA. RNA is just a simple chain, and I will talk about it a little bit here, that does the coding of a protein, which is antigen, that is needed to produce the antibodies. We can put that directly in the body, and there we have antibody produced at a very large scale in a very short time. Now, not that I want to teach today about DNA, how it works out, okay, but DNA goes into RNA and RNA goes into mRNA, which is just a printing machine instructions. It's just a piece of chemistry in an RNA, and it produces an amino acid chain, which then folds up to become a protein. I think this reaction is what has changed what we call a new field. It's called synthetic biology. I would like some of the younger people here, okay, to pay some more attention and see if we can do a little bit more about the future drugs and future therapies using synthetic biology. So how does it really work? Okay, so let's take a look at that. Okay, what happens if you look at on top here? Okay, we have RNA vaccine given to you, and it is folded in a lipid, which makes it stable. And this is what this RNA looks like. Okay, this is just a piece of chemistry. It's got a coding region, and then it's got a tail and the top. And you don't have to go into detail, but it is essentially just a piece of amino acid chain. Okay, that's what it is. So we start with that one, and so if we administer that, this whole lipid, the lipid is needed to protect RNA because it's extremely unstable. And this lipid is mostly a nanoparticle, which is easier to put inside the body cell. Now, keep in mind, that RNA vaccine does not go in the nucleus. All the work it does is in the cytoplasm, which is what makes it safe. So it cannot affect your body DNA. So, and then it dies off very quickly. So it goes in the body, in the cells, and ribosomes, these are the machine that takes the coding from RNA and converts it into proteins. So it makes the proteins. And then I don't think I want to go in detail what happens afterwards. All I know, or I can tell today, is that these proteins now body sees as if they have come from a virus. That's it. Everything else that happens, all the reactions, they call MHC1, 2, I don't think we need to worry about that. All you need to know is that now we have an antigen only. Now, this antigen or this protein is not harmful at all. It cannot do anything. It's not virus that will give you the coding to multiply. So the way coronavirus works, okay, it has a DNA code. Once it gets into your body cells, it tells the cells to reproduce it. And the body sees this as, okay, that's a good instruction, I'll reproduce it. And it keeps reproducing until the cell bursts, and then it spreads all over and you stop breathing. So RNA vaccine simply puts a part of the antigen in your body cell. So your body reacts as if you had an infection and produces all kinds of cytokines and antibodies needed to take care of it when it actually happens. The dosing of RNA is very small. I think this Pfizer vaccine has a dose of about 20 microgram only. So you don't have to make a lot of it. Okay, if you can make 1 kilogram, you can produce 30, 40 million doses, which could be worth hundreds of millions of dollars. Okay, so this is a small scale technology, but nevertheless very well proven. By the way, the flu vaccine yearly dosing is about 200 million doses around the world. So how do we make it? So I'm going to tell you how to make it. It may seem like a simple process because it is simple. So you take a virus, you chop it up, okay? Then you take out a protein on the surface of it, and you analyze it and sequence the protein. So this is the protein I want to make. Now, once you have the sequence of the protein, by the way, Mrna got the sequence of protein from NIH for COVID virus, and in 4 days, they had the vaccine ready to put into humans. That's how fast this process can move. Then you will see the next step that is grayed out, but that's the standard step, okay, which I'll tell you why I gray it out. So now once you have the sequence of protein, okay, you can easily convert that into transcript into what DNA molecule will be needed to produce this protein. Okay. Then you convert that DNA, and then you synthesize the DNA. First of all, you synthesize the DNA, and then you make additions to it in a way to make it resistance to one of the antibiotics. And I'll tell you why, okay? Because once you are going to put this DNA into bacteria, not every bacteria will take it, okay? So it's just to differentiate the bacteria which are useful and when they are useless. So you go down in a bacterial plasmid. Plasmid is the DNA in a bacteria. The DNA just floats around. It's a circular DNA that floats around, and you take the DNA out and paste your DNA in it along with few makeup items, and then you put it back in the bacteria, and the bacteria begins to multiply and start producing millions and millions of copies of the DNA, your DNA that you want to produce. Once you have a boatload of DNA, then you go and remove it out there, straighten out the DNA, and you modify and convert it into RNA, which is very simple enzymatic reaction. And then you add a couple of things. You add a cap and a tail. These are all specific, very easy chemical reactions to make it work in humans. Now keep in mind, this DNA has to, or this RNA has to be recognized by the body that it is indeed a foreign RNA, foreign piece, and that it has a start and ending cycle, what to do with it. Then there's a lot of purification that goes on. Now, going back to that shaded part of it, you can also avoid all of it, okay, by a new process which is coming in now. It's called PCR. Why not just simply take the DNA that you like and make billions of copies? It is possible, even though the cost of technology is not suitable today, but I'm just letting you know that maybe in the future, we don't have to go through all this plasmid DNA and bacterial production. Once you have RNA, you purify it because there's a lot of things in there that are not desirable, which, by the way, will not be if I were just doing a PCR reaction also. Then you wrap it up into a lipid because you want to protect it. And this is a nanoparticle because you need a very small particle so it can enter through the surface of the cell. Then you do all your testing. You do animal efficacy, animal toxicology, phase 1, phase 3, and before you know, you're ready to give a vaccine, COVID vaccine perhaps, okay, to hundreds of millions of people. So going back to, again, very quickly, not that I want to emphasize this, but I wanted to make it very simple that plasmid vector that goes into bacteria is known for a long time. It was very relatively easy to make. There's nothing in there. There's no art or science involved that is not very well understood. And then here is the two processes that we talked about. One is the linear RX, which is the PCR process, and the other one is a plasmid process. And you can see the big difference between those two processes. But here's the part that I'm very excited about. Okay, so you have mRNA vaccine. Generally, vaccines are used, as the name says, but somehow vaccine doesn't mean it is only for infections. It's to prevent something, but mostly it is considered as a product to prevent infections. My idea is, why not use it to prevent diseases also, other diseases? But before we go that far, one of the biggest advantage of this mRNA vaccine is that you can take multiple RNAs, different diseases, and you combine them together and you have single vaccine for maybe for 10, 15, 100 different infections. Never was it possible before even to think about it. Flu season comes. Every year we get a new vaccine because the virus mutates. Now, in these vaccines, we can continuously change the amino acid sequence in the mRNA to produce an antigen which will be representing the current mutation. I'm sure you all heard that the coronavirus has gone through mutation. And while the current mRNA vaccines may still be effective for the mutation, I'm totally confident that within the next few weeks, these vaccines will be updated. They will call them updating with a new mRNA that will be effective for the new species or new mutation of bacteria. Never was it ever possible to do that. But now we're talking about applications which are far beyond infections. How about if I produce an antibody that will block up the antibody in our body that attacks the diabetes cells? The rogue antibodies, I call them. I can treat all autoimmune disorders, Parkinson's, Alzheimer's, diabetes type 1, actually prevent them from happening. I'm not providing a treatment because there's no treatment, but I can certainly prevent them. Imagine if I can give a vaccine at an early age for all the STDs, papillomavirus, HIV, when children are about 10 or 12 years old. These diseases will be gone forever. So the point I want to share with you is that the impact of this vaccine will be so large on humanity, and not only in preventing diseases, but also controlling the mortality rates, which is one of the biggest issues now in Pakistan. So this is indeed the future. So what does Pakistan need? Okay, I don't want to be a commentator here sitting 10,000 miles away and tell you what you need to do in Pakistan, but I am from Pakistan and I have continued my association with it. So I cannot leave you without saying a few things. Okay, Pakistan has the 8th largest population. We're pretty good at making humans. Okay, there's no dearth of it. But we are 154 out of 195 in healthcare quality, everywhere. I think this is one of the worst statistics of any country I've seen. It's about time we do something about it. First, we had botanical drugs. When we came out of the ice age, all we did was we ate whatever was available on the trees and the bushes. Some of them worked, some of them did not work. And that was only 10,000 years ago. Then about 200 years ago, the chemistry came in from Germany and we started making chemical drugs, mainly from botanicals. And then about 50 years ago, we entered an era of biologic drugs, basically going back to your body, okay? What helps us survive for millions of years, okay? But not any of the chemical medicine or any of the diagnostics, but what we have inside our body. And now we're into genomics, okay, where we're actually modifying the body's genome. We are replacing the proteins in the body that body stops making, such as insulin. But also now we are moving forward to gene therapies and other things. Pakistan is still struggling to make safe chemical drugs. When I say safe chemical drugs, okay, please don't take me wrong, because I'm always blamed for making such comments, is that none of the drugs made in Pakistan is ever allowed to be sold in any country where quality matters. Why would they not sell it? There's a lot of money to be made. No further comments. There are no biological manufacturing in Pakistan, period. There are few archaic vaccines made by NIH, and I don't know what they're doing for commercial production. They should be in the hands of private entrepreneurs. Now, you should know, by you, I mean, if someone outside there who has motivation to move forward, it doesn't take a lot of space and investment to go into biologics, as wrongly perpetrated, because small facilities you need more science. Think of this way: your neighbor that you keep fighting with all the time is the largest manufacturer of vaccines in the world, period. We already got 2 COVID vaccines. They sell billions and billions of dollars. Even Bangladesh is selling billions of dollars of pharmaceuticals to US and EU. Not a single dollar goes back to Pakistan. The point I'm raising is that it will take a private sector initiative. And I think NED, with all of its expertise, can form some kinds of consortium to educate the industry why they should move up and get into biologics, get into mRNA vaccines, get into the science of the future to improve our statistics from 154th out of 195. I like to see it in the at least no more than 50 or maybe better. And then you can also tell them there's a big market. Look at this HPV vaccine market. HPV is $12 billion vaccine and growing very fast. Same thing happens, all these autoimmune disorders, the drugs used to not to treat them, but to support them are into billions or hundreds of billions of dollars. So there's also a commercial possibility. And I'm really hoping, I'll close it with this slide, that I really hope that Pakistan will rise to the challenge of COVID-19. I'm very sad to see that there's no vaccine being made or a good vaccine made available that can be affordable. I have given my views to the health ministry about which is a safer vaccine as a member of the FDA team. And I truly believe, there is a lot of opportunity for us to do something very good for Pakistan. And I think RNA vaccines manufacturing will be one of the areas where I will recommend we look into it. Thank you very much. Assalamu alaikum. Stay safe. Khuda Hafiz.